And again, I know you all know what a nucleus is. The bacteria don't have it, but we do. Everybody else does, except bacteria. Now this one you might not know, I'm not sure. The nucleolus. The nucleolus is found in the nucleus, and it makes ribosomes. Now I would normally ask you a question that anybody know what the ribosomes do. Unfortunately, it's right at the bottom of the slide, so you can look and see what it does. The ribosomes make proteins. The ribosomes make proteins. So one more, and then I'll show you a picture. The cytoplasm. Cyto means cell. Cyto means cell. The word plasm just means material. So cytoplasm is literally the cellular material. It's where all the organelles are found. So that whole thing there is the nucleus. Right there is the nuclear membrane. It's a lipid bilayer, just like all membranes are. This big old black thing is the nucleolus. If you can't find the nucleolus, you will be back here next year. It's pretty hard to miss. And then outside is the cytoplasm. And the cytoplasm will go all the way out to the cell membrane. So with that, let's talk about some of the actual organelles. Starting off with the mitochondria. Now again, I very much assume you all had that before somewhere else. The mitochondria is always called the powerhouse of the cell. That's because it makes the energy for the cell. Where does the mitochondria get the energy from? That is the energy, but how is it making it? Where's it coming from? Glucose. Glucose. The mitochondria breaks down the glucose to produce energy in the form of ATP. Pardon me? Well, the plants have chloroplasts, and the plant will get energy from the sun. Yeah. She was asking if plants also have mitochondria. Actually, they do, but mostly it's chloroplasts. So this is a picture. ISTAE. I'll put it on the board. But those lines there are extensions of the mitochondrial membrane. So you can almost see the lipid bilayer, almost, going around here. That's the membrane. And these cristae those cristae are extensions. The reason I mentioned that is where the glucose breaks down. That's where the energy is produced.
In the first.
Mm-hmm. And there are other types of mitochondria, but basically they all do the same thing. They make energy for the cell. But there's also something else that's really pretty cool. And again, I'm pretty sure most of you are familiar with mitochondria from other classes, biology and so on. But this other part, I'll bet you're not familiar with it. The mitochondria are the only organelle, besides the nucleus, that has its own DNA. No other organelle has its own DNA. Now the reason is because the mitochondria are considered to have originally been bacteria. They're originally considered to have been bacterial cells, and then way long ago, in early days of evolution, the early bacterial cells merged with what became our cells. And the mitochondria produced ATP for the cell, and the cell gave the mitochondria food, glucose. So, because they used to be bacterial cells, they had their own DNA, and they still do. Now, the relevance of that is that the mitochondrial DNA will pass down generation to generation, just like normal DNA is. However, normal DNA, as you well know, comes from both parents. Every single one of us is a mixture of both parents, our mother and our father, not the mitochondrial DNA. The mitochondrial DNA passes down unchanged only through the mother. So every one of us has an exact copy of our mother's mitochondrial DNA. So male or female, we all have an exact copy of our mother's mitochondrial DNA. She, of course, has an exact copy of her mother's mitochondrial DNA. So what that means is we all have a copy of our grandmothers and great-grandmothers, all the greats, all going back only through the female side of things. There's a reason for that, by the way. Off the record again. Now, obviously, you all know what you do. The answer is not family history. That's not what we're after. So if that comes up, the answer is the mitochondria make ATP. They make energy. But I did want to present that to you because I think it's very interesting. Yeah?
You go back far enough, aren't we all from Africa?
Yes.
Couldn't we all be there?
Too far. Remember mutations every 300 years?
Okay.
Yeah, she was asking, well, are we all from Africa? Yes. The thought is, well, the original, we're really off topic here, but doesn't bother me. The birthplace of ATP, they make energy. So let's go ahead. The ribosomes make proteins. I'm sure you have that already somewhere. The endoplasmic reticulum, the ER. Endo means inside, plasm means material. The word reticulum, which you'll see again off and on during the quarter, means a network or a system. A network or a system. So in this case, it specifically means a network of internal membranes. So it's not the cell membrane, it's internal membranes. And there are two types of ER, rough ER, smooth ER. The rough ER is rough because it contains ribosomes. Now notice I'm saying it makes proteins. Yeah, I didn't know how to say that, and I still don't know how to say it. What's making the proteins are the ribosomes. The ribosomes are in the rough ER. So technically, the rough ER is producing proteins. But really, no. The ribosomes are making proteins. So if I were to ask you what makes proteins, the answer is ribosomes. If you try to say, well, it's rough ER, I'll mark it wrong. If you try to say, well, I have it in my notes, I'll say I don't care. It's the ribosomes that make proteins. The ribosomes are in the rough ER, so I'm sure you can see where I'm going. I just didn't really know how to express it properly, and I still don't. So it's the ribosomes in the rough ER that are making the proteins. What the ER actually does do, it makes phospholipids, and it makes cholesterol. Now, that might surprise you a little because cholesterol, of course, has a bad reputation. But in fact, we use cholesterol. Sorry. It's actually quite a legitimate molecule. And you're wondering now, where am I coming with all this? The problem is not the molecule itself. The problem is having too much of it. Then we get in trouble. The cholesterol itself is used to make steroid hormones. So we use cholesterol. You just don't want too much of it. So at any rate, The ER itself will make cholesterol, will make the phospholipids. The ribosomes in there will make proteins. ER is smooth because no ribosomes. And it does make lipids, it does make steroids from the cholesterol. Questions so far? I know it's not the most exciting thing in the world, but we'll get through it and then we'll get some better stuff. Okay. Golgi apparatus. That's named after Mr. Golgi. Another system of internal membranes. This one packages proteins. So I need to explain that. Actually, I'm going to explain it by going back to the rough ER. The proteins come out of the ribosomes in the rough ER. And then, before actually being released, the rough ER will do some packaging of its own. Now, the packaging of its own means to add sugar molecules. The rough ER will add sugar molecules to the proteins. Well, sugar plus protein is being converted into a glycoprotein. Remember, glycoprotein can be information molecules. Also used for stickiness, but in this case, information. And the information being added is acting as an address label. Acting as an address label. The glycoprotein, the information, tells the glycoprotein to go up to the Golgi apparatus. In the Golgi apparatus, further packaging is done. The packaging is adding more sugars, taking away some sugars, probably doing both. Probably doing both. And then the Golgi apparatus will give it its final destination. Usually outside the cell. Usually for export. So again, proteins produced in the ribosomes in the rough ER will actually have sugar molecules added to the proteins, making them into glycoproteins. The information in those glycoproteins sends the molecule to the Golgi apparatus. The Golgi apparatus does more labeling, more packaging, and so on. That gives the glycoprotein its final destination, probably outside the cell. It's a little bit like mailing a letter. You can put your letter organelles, some other organelles. Lysosomes. These guys are garbage collectors. The cell is often compared to being a factory. And in being a factory, well, it makes products, but it also makes waste products. Every cell does. So something has to come by and pick up the waste products. Lysosomes do that. Lysosomes are little sacs of digestive enzymes. They're little sacs of digestive enzymes, and they pick up the garbage. They also contain HCl, so they break down stuff. Sometimes the stuff can be recycled, and sometimes it's just garbage. It'll be sent out. So lysosomes are the garbage collectors, the janitors, whatever you want to call it. Microtubules, microfilaments make up the cell skeleton. Essentially, the cells have little bones just like we do. Now, they're not really bones. Yep. They're not really bones. They're microtubules, microfilaments. But they give the cell size and shape and structure, just like our bones do. Cilia and flagella. The cilia are used for transport. The flagella used for locomotion, for swimming. Let's do the flagella first. Many bacteria, but not all, many bacteria have flagella. For humans, there's only one flagellated cell. Unfortunately, I have it on the board right behind me. So that's it. That's the only flagellated cell that we have. Bacteria might have flagella, they might not. They don't all have it. But they're for swimming. They're for locomotion. Cilia little hair-like structures we have lots of ciliated cells, lots and lots and lots. In our respiratory tract, our digestive tract, reproductive tract, all over the place. And all these cells are together, and the cilia are all kind of moving together. So basically you have a cargo, like for humans you'd have mucus or something, and the cargo is carried from one cell to another, like a conveyor belt. The cells themselves don't move, but the cilia do. And the cilia just kind of move things along, like I say, like a conveyor belt. A better example, but I'm not sure all of you have ever seen it, if you ever go to Husky Stadium, the students will get plowed quite quickly. Well, cilia and flagella for bacteria are used for locomotion and for transport. This one we're not going to do. I use these same notes for my A&P class, so not all of it applies to us. The reason it does not apply to us is because it involves mitosis. Bacterial cells do not do mitosis. Why not? Go ahead, what? Perfect. They don't have a nucleus. Mitosis is division of the nucleus. I know everybody thinks of it as cell division. Not quite correct. It's division of the nucleus. Bacteria don't have a nucleus, therefore no mitosis. What they do for cell division is called binary fission. And we'll talk about that. We're not going to get very far into this, but diffusion. Now we only did a little bit of cell transport. We talked about the recognition proteins and receptor proteins, but those are really for individual molecules. Really for individual molecules. These are for larger quantities. Now diffusion, frankly, is something you're familiar with. Diffusion is moving from a high concentration area to a low concentration area. Now examples would be dropping food coloring into water. If you drop food coloring, at first everything's just in one little spot and then it starts spreading out. That's diffusion. Or if you're cooking in the kitchen, well, the aroma will start in the kitchen and then it spreads out all over the house. That's diffusion also. So, we've all done it.